Hello, today we're going to be talking about DNA point mutations that can alter the amino acid chain being produced. Here we are given a DNA sequence, and we need to identify which strand is the template strand and which strand is the coding strand. Remember that RNA polymerase reads from the 3' to 5' direction to produce an RNA strand in the 5' to 3' polarity during transcription. Also remember that RNA replaces every thymine with uracil. After scanning the DNA strand, RNA polymerase will have found the start codon AUG, and this will establish our reading frame until it reaches the stop codon. Codons are triplets of bases that are used to produce an amino acid. If we look at the bottom strand and then read backwards, we see that we have a start codon and stop codon, and between them we have 3 more codons. This tells us that this is the coding strand because it matches the RNA strand, with the exception of thymine, and the top strand is the one being transcribed, so it is the template strand. This makes sense if we think back to the template strand being transcribed in the 3' to 5' direction, with the product being in the 5' to 3' direction as the coding strand. Here I have flipped the coding strand reading from the 5' to 3' direction, and the RNA strand that is produced reading from the 5' to 3' direction. Again, here are the start and stop codons. Based on the RNA strand, these are the amino acids that are produced. They are methionine, valine, serine, lysine, and the stop codon. This is our original 4-chain amino acid sequence. Our first type of point mutation is missense mutation. I have the original amino acid chain in the top left, and the original RNA strand in the top right. These will appear in each mutation, and the blue arrow is pointing at the base that is being changed. In our coding strand, our C was replaced with an A, and this is then seen in the RNA strand. The amino acid produced changes from serine to arginine. The change in the amino acid is what tells us that this is a missense mutation. Also, this is a transversion mutation, which I will explain soon. Transition mutations occur when the change in DNA includes having the same shape. Transversion mutations occur when the shape changes. There are only 2 shapes, and they are purine and pyrimidine. In our missense mutation, we had a change from pyrimidine to a purine, which is a shape change. This gives us the transversion mutation. Our next mutation is the silent mutation. This results when our change in base produces the same amino acid. Here is the change in the DNA sequence, and then here is the change in the subsequent change in the RNA strand. Our new change still produces serine like the original, so this is a silent mutation. This is also a transition mutation because we go from a pyrimidine to a pyrimidine in the change from a C to a T. Our next mutation is the nonsense mutation. Here is the change in the DNA sequence, and then here is the subsequent change in the RNA strand. This results whenever we produce a stop codon earlier than the original stop codon. Therefore, the amino acid chain becomes smaller, and this change is a transversion because of the change in the shape from an A to a T, which is the purine to a pyrimidine. Our final point mutation is read-through mutation. Here is the change in the DNA sequence and then the change in the RNA strand. This results in our stop codon changing into an amino acid, and the chain increases until it reaches another stop codon, producing an even longer amino acid chain. Our change now produces tyrosine, and will have subsequent amino acids attached to the chain. This is also a transversion mutation because we change from an A to a T, which is a change from a purine to a pyrimidine.